Thank you very much. Okay, thank you very much for the invitation. So my name is Daniel Packwood. I'm from an institute at ICES, Institute for Integrated Cell Material Sciences at Kyoto University. I'll be talking about data science for stem cell science and biosensor research. So I'll briefly introduce our institute just to begin. So we belong to a network of institutes with WPI. Okay, that includes ICES, where Corbett used to work, and several other people here work. We're working more on the probably biological chemistry side of things. We work on science on scales between single molecules and single cells. So we have a mixture of chemists and biologists in our institute. The chemists work on things bigger than single molecules. The biologists are working on things smaller than single cells. We work in this mesoscopic domain, consisting very much of self-assembled materials. So if you're ever in Japan and if you want to come and visit us, please let me know. I'll be more than happy to show you around. So I'm the PI of the theoretical group. We do a lot of material science, but today I'll be presenting two of our most recent topics, which are probably a little bit more in the biological direction. The first is metal organic framework sensors, and the other is organic molecules for stem cell control. So let's get into it. Metal organic framework sensors. So we had a wonderful talk, a shocking talk about metal organic frameworks just before. I don't have to say too much about these. They consist of metal ions or clusters of metals and organic linker molecules that self-assemble to form these beautiful porous networks, as you can see here. They're discovered by Professor Kitagawa and a few others in the late 90s. I mention that because Kitagawa used to be the director of our institute. He's also a collaborator for this project. And the properties of these materials can be tuned by using different linker molecules and different metal ions and different organic molecules. And as we know, there are millions of different materials that have been reported, although we know half the materials in the database is maybe a little bit strange. Interesting. So a new direction has emerged in this area by the Kitagawa group. They published this a couple of years ago. Hemi-resistive sensors for nitro explosives. So they made this composite material. It consists of a metal oxide electrode, titanium oxide, okay, as a pillar. And that material is covered in a sheath of metal organic framework, one called NH2-MIL125. Okay, this is what it looks like under TEM. You've got these towers of these metal oxides, titanium oxides, covered in this metal organic framework. And what they did is they tested its chemi-resistive sensor properties. So they took these different molecules and they fed them into this material here. The gas molecules float through to this interface. They interact with the interface, they affect its electronic properties, they affect the resistivity of the metal oxide electrode, and that resistivity change can be used as a basis for sensing these gases. And you'll see they got a really strong response for these nitro explosive molecules, these four here. And they got a very weak response for these other molecules. So based on that, they concluded that this material could act like something like a sniffer dog, can detect dangerous compounds. So very good. What we were considering as theoreticians, the question they had for us was, can we use this for other sensing applications? So we sat down, my postdoc and I, and we thought, okay, well, let's see if we can apply it to early disease detection. You see, human breath contains thousands or over 1,000 volatile organic compounds. And abnormal concentrations of those compounds can indicate the presence of certain diseases. Okay, so it's a real fingerprint for your health profile. So the question is, as theoreticians, can we answer this question? Can this NH2-MIL125 titanium oxide be used as a breath-based disease sensor? That's a really hard question for a theoretician to answer, very hard. For each disease, you have to simulate the transport of gas up to this interface. You have to simulate the electron transfer, the charge transfer through the titanium oxide. Not impossible, but it'd be very hard to do. You don't want to do that in a high throughput manner. However, we can use data science to get some very quick and very dirty insights into the problem. And that's what we'll do here. So here are the contributions of our work. The first is a method for simulating data related to the molecule-sensor interaction. I'll clarify what that means next. And the next part is a statistical method. It's not a new method, it's actually a very elementary method. Some statistics for assessing whether or not a disease might be detectable. So let's get into this. Here's our method for generating data. There's a nice database that has been created by a group in Taiwan, the Human Breathomics database. It contains over 6,000 organic molecules from breath, and it has the diseases with which they are associated. In our method, we just sample from this database. We insert the molecule into an atomistic interface model, and we do a geometry optimization using DFT. And you can see the method we use at the bottom. We compute the binding energy, that is the interaction strength with the whole sensor, and also the change in the density of states. I'll define that more clearly on the next slide. So we end up with a table of data that looks like this. We've got a whole bunch of molecules, the binding energy, and this thing W, the change in the density of states. So just some notes, steps 1 to 3, we don't do for all 6,000 compounds. It does take a bit of time. We do it for a sample of 130. Step 2, this relaxation, that's really slow. I don't have time to talk about it, but we made a cluster-based method for optimizing. You can probably guess what it's about. Step 3, let me talk about that now, this change in the density of states. How do we quantify that? Here's the density of states of this material without any molecule hosted inside. That's the blue line here. Here's a Gaussian function centered at the Fermi level. Here's the same thing again, but I've just multiplied the density of states by that Gaussian function. I've weighted it by the Gaussian function. Here I've done the same thing, but I've put this molecule TMP inside. This is what the weighted density of states looks like. You can see there are some changes at these points here. We've got a new peak appearing nearly just beneath Fermi level. This peak here has grown. These ones have shrunk a little bit. Here's another molecule, acetone. This interacts very weakly with the sensor. And it has, you can see some small changes down here. We can quantify those changes using something from the theory of optimal transport called the Wasserstein distance. We treat these two things as probability distributions, and you can measure how much it changes from here to here. That's measuring how much the density of states changes. It's got this horrendous mathematical formula. Mathematicians say it measures the amount of work, quote unquote, you need to stretch, bend, squeeze this blue curve into the purple one here. It's not mechanical work, it's not thermodynamic work, so don't take it too seriously. It's just the amount of effort you have to use to turn the blue one into these purple lines here. And we go ahead and compute the Wasserstein distance. For TMP, for the nitro explosive, it comes out at 0.07, for acetone 0.03, so we can conclude that TMP causes a greater electronic structure change than to the sensor. So here we're going to validate our data collection method by comparing to some experimental data, the experimentally tested compounds. So these are all, or most of the compounds that were tested by the Kitagawa lab. The blue ones are the ones that were undetectable. The sensor could not detect those. The red ones are the ones that the sensor could detect, the nitro explosives. You see, basically, there's a good consistency. Okay, the detectable and the undetectable compounds are clearly distinguished. Okay, so it seems there's some consistency between our quick and dirty data collection method and the experimental outcome. Here I've done the same thing, but I've plotted those 130 breath compounds. And this is what we have here. These are the small green points, the dirty green points. So you can see that most of these breath compounds lie near the blue undetectable ones, which suggests, not rigorously, but it roughly suggests that these compounds will not be detectable by the sensor. But there's a few compounds that are trying to drift towards this region of high detectability. Here I've plotted the data again, but I'm just showing the breath compounds associated with these 12 diseases. Things like COPD, chronic obstructive pulmonary disease. I don't know what that is, but it sounds awful. Diabetes, hepatic disease, liver disease, all these other things. And this is what we get here. You can see a couple of promising, if I can say that word, promising cases. COPD and lung cancer look like they might be detectable. This tail kind of drifts down to the bottom right. But it's not a very rigorous conclusion. It's not a scientific thing to say. We've only tested a small sample of compounds. So let's do some statistics. Statistics is about drawing population-based conclusions from small samples. Let's use statistics to get some quantitative conclusions. We perform something called the student t-test. It's a very elementary method from statistics. What we do is we imagine that these blue compounds, these undetectable ones, are a small sample from a big population of compounds, most of which have not been tested. Maybe many of them are not known. And these green points, these are a small sample of breath compounds associated with these particular diseases. And we test something called a null hypothesis. The first null hypothesis is that for a given disease, COPD, lung cancer, or whatever, the population average binding energy for the breath compounds will not be stronger than that of the undetectable compounds. The other is that the population average Wasserstein distance for the breath compounds will not be larger than it is for the undetectable compounds. So the null hypothesis is that is that this distribution of the green points is not more drifting down to the bottom right than we have the undetectable ones. And just adopting some statistics conventions, if this thing, the p-value that we calculate is less than 5%, we reject these null hypotheses. Now we go through and calculate these p-values. We find that most of the diseases, 7 of them, we cannot reject those null hypotheses. That means that it's pretty unlikely that this thing's going to be able to detect them. Okay, that's a conclusion I'm pretty confident with. Okay, just from some statistics, we can get that. You can see though, there's a few diseases, particularly COPD and lung cancer, for which the p-values for both binding energy and the Wasserstein distance are somewhat small, which suggests they may be good targets. It suggests we've got very good evidence that these null hypotheses are not true, and that they could be targets for further experimental work, which would involve some clinical trials, I think. So that's as far as we've got with this project. It's just purely theoretical at the moment, but this is giving some clue as to what the next step should be for this kind of research. Note that the next steps would be very expensive. That'd be a clinical trial. Okay, now the next talk, organic molecules for stem cell control. So stem cells. Stem cells are cells that can transform into any other kind of cell, but you have to add the right chemicals. For example, this is a stem cell here. I'm sorry for the Japanese English combined slide. Anyway, this cell, stem cell, if you add compound A, it'll turn into a blood cell. If you add compound B, it might become a nerve cell. If you add compound C, it might become the cells inside your guts. The problem is no one knows what compound should be used. That's a complete mystery. There's no rational design strategy for making these compounds. Now, what we're interested in is cardiomyocytes. These are the cells that make up the heart, very important cells. You don't have them in your diet. Now, my collaborators, they gave me a small database of 81 molecules. They've done some screening on these molecules. It's very hard to do that screening. And they had 26 of which, 26 of these compounds could induce cardiac differentiation. They can make this cardiomyocyte. They had structures like this thing here. And the question was, can you use some machine learning statistics to discover some other compounds? And that's, I said yes, because it looked like interesting stuff, but that's not like an easy thing to do, is it? You've only got 81 molecules. It's a very small sample. And how far can we get? Well, let's think about what we should be doing with small data. This is the flow. I'm sure all of you are familiar with this flow of machine learning. We've got labeled data. We've got to make some feature vectors or descriptors, if you prefer that word. We've got to make a regression model, and we have to go ahead and predict the labels. When you have small data, we're limited to very simple models. Doesn't mean you can't do machine learning. Anyone who says you can't do it is wrong. Okay, it just means you're limited to simple models. Okay, this thing has to have a very small number of parameters. To compensate, that means what's going into your model has to be very rich. It's got to have the information which is very closely related to the chemistry involved in the cardiac differentiation process. You have to choose those features very carefully. The more overtraining obviously needs to be checked as well. So these are 3 things we've got to think about. So firstly, let's talk about these feature vectors. What kinds of feature vectors should we be using? Well, I just sat down and thought about this. I made a hypothesis. The shape of the molecule should be very important. If you talk to any biochemist, they'll say that the molecule shape is important for determining its biological activity. Why is that? Here's the shape as calculated from an CEMDI simulation. The molecule will probably fit inside of some protein inside of the cell. And the shape of that protein will determine the shapes of the molecules that it will interact with. Once it slots in there, it will probably cause cardiac differentiation. We don't know for sure, but it's likely that that's the case based on biological intuition. So the molecule shape should be considered as input. And as physical chemists, we have some intuition for that. It should be the van der Waals exclusion volume. I just call it van der Waals volume here. So let's just keep this first point in mind. Let's go to the second point. The other thing that biochemists will tell you is that hydrophilicity should be important. Okay, so what does that mean? How do we incorporate hydrophilicity information into that van der Waals exclusion volume? Well, I made up this little procedure. We break the molecule up. We just chop it up at the rings, and we add hydrogen atoms at the broken parts. We put our molecule into virtual water. We do an CEMDI simulation. We calculate hydrophilicity as the interaction energy between that fragment and the water molecules, after normalizing by the number of atoms in the molecule. And then we just kind of paint our exclusion volume with those hydrophilicity values. So these orange parts are the bits that have low hydrophilicity. The blue and green parts, especially the blue parts, are the bits with the very high hydrophilicity. So these are the two considerations we have. So we go ahead and compute these decorated van der Waals volumes for our molecules, and we go ahead and do this procedure. This has been reported in the computer graphics literature. It says that you can expand any function in terms of a basis of these functions Z here, which is a product of a Zanicki polynomial, that's this R, and this thing Y, spherical harmonic. And that's a result from mathematics. Then we can compute these things here called Zanicki moments, capital F, little nl, which is just a combination of these coefficients here. They have a really nice property. Those are rotationally invariant. If you rotate your molecule, the value doesn't change. So that means it's a very good descriptor. We don't have to worry about our molecule's orientation in our data set. But we can make a feature vector just by computing a whole bunch of these Fs for the different values of N and L. We take a thermodynamic average over 10 molecular dynamic snapshots. We take those snapshots and relax them and get the value of F. And if you use N equals 25, you get a 351 dimensional representation. But don't worry, there's huge amounts of correlation between those Zanicki moments. So you can do some PCA and just get rid of the unnecessary ones. You can get down to a dimensionality of 7. Okay, so what kinds of results do we get? We're going to use a logistic model, a very simple regression model. Very, very simple. It's just got 8 parameters. That's probably the largest number of parameters we can consider with our data set. Here's the performance. Now, first I'm considering the case as a baseline where you don't have any hydrophilicity decorations, just the molecule shape. You achieve an accuracy of 74%. That's not bad. But the true positive rate is pretty shocking, 35%. Okay, you can't really do much of that, especially if you want to do molecular design. But then you add this hydrophilicity information, the accuracy bumps up a bit to 83%. The true positive rate shoots up to 65%. Okay, that's a useful value that we can use for molecular design. So I think this result's showing you that shape can predict the absence of the cardiac differentiation effect, but hydrophilicity information is needed to supplement that information and predict the presence of the effect. This is a closer look at the predictions. I'm plotting the molecules in the 2-dimensional embedding, just using those Zanicki moments as feature vectors. They're clustered according to the molecule shape. I don't have the molecule pictures here, but you can see that it's a relatively good agreement between the experiment and the logistic model predictions. But there's a couple of regions where it doesn't work so well. We just have to keep that in mind when we go ahead and design some new molecules later. Now, overtraining. How can we test for overtraining? This is a really cool thing with these decorated van der Waals exclusion volumes. There's a very natural way of doing it. So we can define overtraining as an excessive sensitivity of the prediction of the model to its inputs. So a very high sensitivity of the prediction of this model to the value of Q here. We can test for a model overtraining by randomly perturbing the hydrophilicity values. So what do I mean by that? So here's the unperturbed case. This is what we have normally, and that's the model's prediction. We just add a little bit of random noise to those hydrophilicity values. We make a whole sample of them, 25, and we look at which ones cause the model's prediction to change. We can't close to 1. Model case. So let's now go. We have a fragment line here. These fragment molecules come from the training data, which therefore means that these molecules should lie close to the training demand the model. That prevents the molecule the synthesis testing. We want to be very careful with the molecule we pick because we're going to go into an experiment now. Experiment. This costs one postdoc and a whole lot of other stuff you have to purchase. So yesterday he did the synthesis. So every time the student did the programming. See here, but these three genes of her cells. Sorry, these cells have these genes fully expressed. It suggests what means that they're cardiac cells. Here's our molecule at low concentration. You can see it achieves a comparable expression to this molecule XAV. XAV is a very famous, very potent cardiac differentiation molecule. So our molecule is not better than the best, but nonetheless, it's getting there, that domain. Here's some images of our cells. Okay, we've dyed the cells using, I can't remember the name of the dye, actually it's written there, DAPI-CTNT. Those dyes, they stick to the fibers inside of cardiac cells. So that's also a further indication that these are in fact cardiac cells. So there you go, the cardiac differentiation effect was observed at low concentration. So if anyone says that theoretical chemistry is useless, we can say we can make heart cells with it sometimes. Okay, that's the end of my talk. I'll just summarize. So in the first part of my talk, I showed a method that we developed for characterizing the molecule MOF semiconductor sensor interaction based on binding energies and the Wasserstein distance. And we used some statistical inference to suggest that COPD and lung cancer might be detectable with that material. And in part two, we developed a regression methodology for predicting the cardiac differentiation effect of organic molecules on stem cells. And it seems to work pretty well on small data sets. And with that, we designed a new compound and confirmed its cardiac differentiation effect. Just make some acknowledgements here. So my worked on the sensor project. Fatima did the experimental testing in the sensor CEMDI project there. And also we have to point out our funding. So thank you very much.
Yes. Yeah, I have some questions regarding your gas sensor.
Yeah.
So you have been doing computation, for example, with acetone or different gas. We know that, especially if you are applying to breath sensing, water will be the nightmare. Like it has to be water resistant in order to be sensed. So
Yes,
you try also to do some modeling on with water to see how the water will affect your electrical property.
Oh, we didn't go that far. You mean how to mean actually doing modeling of the actual true sensor the true sensor result? Is that what you mean?
No, even by changing your charge, the density of state, because the sensing property will definitely differ depending on the environment.
Yes.
And if we test only the gas,
Oh, right.
the radius, if someone is breathing on the
Yeah, because you have water in your breath.
you're worried, yeah.
Yeah.
And the humidity of one of the nightmare of this gas sensors. So
We didn't do that, no.
Okay.
It's very special breath that has no water.
Yeah.
Yeah, but that's a really good point. I think next time it'd be better to put water in the sensor, in the model sensor.
Maybe
Yeah.
to see how the charge density will also affect the update.
Exactly. And it might even mask the changes in the density of states.
Okay,
Yeah, it's a great question.
thank you.
Thank you.
When you compute stress associated with the change in the density of states, you're basically computing the change in the band structure particles of energy,
That's right.
which could be completely swamped by changes in other particles of energy.
Hmm.
Which so that sounds like a negative thing. On the other hand, it would suggest that even though the band, even if only the band structure particles of the whole energy is already useful, then maybe there is an even more significant pickup.
Hmm.
Right, to start looking at changes in other parts of it.
All right, right. So look, okay, we haven't looked at other changes in the materials, but you're suggesting that other properties related to energy should be looked at, perhaps.
As a component of total energy.
Right. Okay. Yeah, we can look at that. We haven't gone beyond what I've shown here. Yeah, that's a great suggestion.
I also have a question about the Prof sensor study. So you mentioned that you had to reject the null hypothesis for several different compounds.
That's right. Yes.
Have you considered at this stage using other statistical testing, because the t-test is susceptible to sample size, sensitivity,
Yeah.
as well as the normal distribution of data?
Yes, it is.
So
Yeah, the TCS is good for small samples.
Mm.
I considered firstly, actually, before I did the t-test, we used the Kolmogorov-Smirnov test to compare the entire distributions. But I got similar results, but then I looked more carefully into the theory and I thought we can't really justify these tests, the sample sizes are far too small. Yeah, so I think t-test, I settled on t-test at the end. There's another one, like a two-dimensional hotel test. That's another one we could consider. I couldn't find a good hardcode for doing that one, but sure it exists. There's many tests you could do, I think. Yeah, I also looked at a analysis of variance, but also again the data was too small for that. Yeah, you can apply a whole battery of these tests if you've got the right amount of data. Yeah. Yes.
Can we go back to the slide with the t-test results?
Yeah.
Just,
We're going to push this many times. Yes. It's good exercise.
by the way, I really enjoyed so we have 12 of these, but there's a few of these where the P tells you these are like 1 in 20, but so this would also kind of expect this by chance. Like there's a few of them which are very small, but like for example, COPD is P of 0.04 when you're running 12 tests is almost expected.
Oh, what do you mean? 12 tests? Yes, they got these 12 tests.
Yeah, we're running 12 t-tests
Yeah.
independently, and one of them hits at
Oh,
one shot side out of 20, which is almost.
I yeah, well, it happened at this particular one. I don't have a good response there to think about it. Yeah, I think you've got a good point. Yeah,
We'd like to have a test which allowed you to do a joint.
yes,
Normally, you need to do a club approach correction. You need to be some stricter in what you value set.
right, yes,
Based on the number of things that you've tested.
okay.
Go.
I think I remember learning that when I was an undergrad. Okay, I'll try it, if not more for any direction. Okay, that's a really good point, thanks. Yeah. Okay, any other final questions before we head off for lunch? Okay, thank you so much. Thank you.